Hello and welcome everyone. I'm Simranjeet Maan and this video is going to be very crucial for all of you. Why? Because with this video we are going to start our 10-year UPSC PYQ marathon sessions. Okay. Now you all know the importance of PYQs, but still you keep on delaying it, you keep on ignoring it. Even if you cover it very late, and that too very superficially. Superficially means you know the answers, but you have never analyzed your PYQs. You have never understood the concepts, the themes, the pattern that UPSC keeps on repeating. Okay. And the best way to do PYQs is do them first at the start of your preparation. Why? Because once you do them at the start of your preparation, first of all, almost 60% of the concepts that UPSC is going to ask are already covered in PYQs. Okay. Rest 40%, which you are going to cover through your books, through your current affairs, there also PYQs are going to help. Because once you have gone through PYQs, you have understood the mindset of UPSC examiner. You have understood the taste of UPSC examiner. You have understood the pattern that they are going to use. You will exactly know what to focus on, what to ignore, what to stress on, which facts you can ignore, which facts are important, which concepts are important. Okay, so rather than delaying it till the end and waiting for somebody to make a video, and that to a superficial video in which only the answers are provided, please do not do that. Be an expert. If you have been unsuccessful in prelims, there might be a reason for it. You might be doing it not the way it is desired or not the way it makes you successful. Okay, and PYQs is something which should not be ignored at any cost, and that is why we will be covering all the 10-year PYQs, including CSAT. In this class, I will start with science and technology. Last 10 years, biotechnology and health, I am going to cover, and that too in depth, explaining every single concept. Okay. In the next 3 videos, I will cover rest. Okay. I have divided science and technology itself in 4 videos. So let us start. The first question. Now in 2025, the first question was, with reference to monoclonal antibodies often mentioned in news, consider the following statements. They are man-made proteins. They stimulate immunological function due to their ability to bind to specific antigens. They are used in treating viral infections like that of Nipah virus. Okay. The question is about monoclonal antibodies. Now, antibodies, you need to understand, are part of our immune system. Antibodies are something that are released by our immune system either to deactivate an antigen. Antigen means a foreign particle that has entered our body and is treated as dangerous by our body. Okay, they are like the fighters that fight against those foreign particles. Okay, but they are not the fighters in the sense that they kill this antigen or the foreign particle. No. Antibodies, what they do is they either mark that antigen so that other immune cells which do the work of killing the antigens or the foreign particles can identify them, or they deactivate the sites which actually make those antigens lethal to us. Okay, I will give you an example. Understand this. You must have heard about spike protein a lot during coronavirus. Now think what was spike protein and why it was so important. This was a question asked by UPSC, I think in 2020 also, with respect to ACE receptor. Now speciality of the spike protein was that it was used by coronavirus or the COVID-19 virus as an entry point into our cells. Okay, that spike protein used to bind with the ACE2 receptor. Okay, now the way that our body would fight with those the coronaviruses was that it will release antibodies that will block these spike proteins. Okay, but this was the natural way of doing that. But we understood that this is the way we can deal with this. So what we did, we started developing for multiple different types of viruses. We started developing these antibodies in labs or in this clinical research laboratories. Okay, so monoclonal antibodies. Monoclonal means we have cloned the antibodies. Just give me 1 second. We have cloned. Monoclonal means we have cloned a given antibody and created many copies of it. Okay, so this makes them what? Man-made proteins. Okay, now as I said, already said, and I will show you also, this is the spike protein that I was talking about. This is the case of coronavirus. Now this monoclonal antibodies, what it does, it attaches itself to this spike protein. As I said, now this spike protein was the entry point of this coronavirus into our cells. So once this was blocked, this became neutralized, means the threat of coronavirus was neutralized. So the second point is they stimulate immunological function due to their ability to bind to specific antigens. Okay, so this was correct. This is again correct. Now this one is factual. They are used in treating viral infections like Nipah virus. Definitely they are used in viral infections, but the fact that they are used for Nipah virus is something that has to be learned. Okay, so now you understand the role of antibodies in our body, and you know the role of monoclonal antibodies, that is the antibodies that are created by us in the lab. Okay, and this is actually created for Nipah virus also. So this makes D as the answer, that is all three correct. Okay, now let us move on to the next question, which was asked in 2025. Consider the following statements. No virus can survive in ocean waters. No virus can infect bacteria. Again, this is a repeated theme, exact repeated statement. So no virus can survive in ocean waters, no virus can infect bacteria, no virus can change the cellular transcriptional activity in host cells. Which of the statements are correct? Now we are not going to focus on too much on elimination, because that is something that we do once we have sufficient knowledge. Now we are going to develop our knowledge base. Okay, and you will see that this statement is actually a repeated statement from previous years. Okay, so if you know such statements, if you know these concepts, it will be very helpful in next year also. So first one is no virus can survive in ocean waters. Now 70% of our earth is covered with ocean waters. If this would have been the case, then we would have already used ocean waters to create something which we call antiviral or some viral kind of therapy, but that is not the case. Viruses do exist in ocean waters, so this statement is incorrect. No virus can infect bacteria. This you must know that there are viruses called bacteriophages which actually infect bacteria. So this is again incorrect. No virus can change the cellular transcriptional activity in host cells. Now this is what I'm talking about. If you have done your PYQs, there were questions or there were this concepts related to transcriptome, transcription and all that. Now what is this? No virus can change the cellular transcriptional activity in host cells. Now you need to understand, first understand what is virus. What is virus? Virus is nothing more than a piece of information. In isolation, virus can do no harm. Virus needs a host cell. Virus has its information, its genetic information, but it needs host cell for replication. It does not have the mechanism to produce energy or proteins necessary for replication. So what it does, it enters the host cells. Okay, once it enters the host cells, then it uses the machinery of that host cell. Like if the virus enters inside me, it will use one of my cells. Let's say coronavirus attacks my respiratory system and it enters into my let's say pulmonary cells. There it will do what? It will use the mechanism of replication of my body, means the energy source from my body, the proteins from my body, but with its own information will replicate itself and then do harm to me. Clear? So this statement is again wrong. This is actually what many viruses do. Viruses change the cellular transcriptional activity in the host cells and then they replicate them. Okay, clear? So this is again incorrect. So all of these are incorrect. Answer will be D, none. Okay. This is what I was telling you. The virus is entering. It is now viral RNA enters the nucleus where it is replicated by the viral RNA polymerase. Once it is done, it releases itself from the cell and start affecting other cells. Okay. So these two were the questions from 2025. Let us focus on 2024. In total, in last 10 years, there were roughly 40 questions. Okay, so 4 questions per year just from biotechnology. 4 questions per year just from biotechnology and health. Okay. Now this was, which one of the following is synthesized in human body that dilates blood vessels and increases blood flow? Options were nitric oxide, nitrous oxide, nitrogen dioxide, nitrogen pentoxide. Nitrogen dioxide, you know, this is a pollutant produced due to consumption, this combustion of hydrocarbons, so this will be wrong. Nitrous oxide, you know, it is an anesthetic, so this will be wrong. So either this or this D, nitrogen pentoxide or nitric oxide. Answer here is nitric oxide. Now, first let us understand what is this. Dilates blood vessels and increases blood flow. Now, this is a vasodilator. Vasodilator means nitric oxide is something when released, it leads to dilation of our vascular system or parts of our vascular system. Vascular system means our veins, arteries, vessels through which blood flows. Okay. Now, why it is needed? Because it is there are occurrences or there are instances where our body needs to reduce the blood pressure. It needs to reduce the increase the diameter of the vessels. Okay. For that, the substance that is there is nitric oxide. Okay. So it is not going to be nitrous oxide, it is not going to be nitrogen dioxide, it is not going to be nitrogen pentoxide. Answer will be nitric oxide. Okay. Now let us see. Next one was, in which of the following are hydrogels used? Now this is important. Understanding of hydrogels is very important. Otherwise, we make the same mistakes which many students are making, and it is not their fault because they have been told by many people that whenever there is a question related to application in science and technology, and this was really confusing, to be honest, for many of us also, we thought that answer is going to be D, 1, 2 and 3, because hydrogels is a budding technology. It might be used in lubricants, it might be used in air conditioners. But actually, if you understand hydrogels properly, if you want to imagine, you can imagine them as sponges. Sponges, or what we say, something which can hold a liquid inside and then release it in a controlled manner. Okay. So something with that property will be used for what? Controlled drug delivery. Okay. We will not only transport that material towards the tissue where it is needed, but also we will be able to control the amount of drug that we are delivering at any point of time. Okay. So hydrogels were basically this. So answer would be only 1, because still it is not used in air conditioning, neither it is used in this industrial lubricants. Mostly here we used compressed gases, here we used hydrocarbons. So answer was A, 1 only. Okay. You can see here hydrogels, how they hold the liquids in their mesh structure and they can release them in a controlled manner. Okay. 39th question was another important one. Membrane bioreactors are often discussed in the context of. Now, membrane bioreactors, you need to understand, if you go through the option, you can clearly identify that the answer is D. Why? Because it is talking about membrane, it is talking about bioreactors. Okay. And most of us know that water treatment in our sewage plants or in our water treatment plants takes place through organic and inorganic processes in which we break down the organic and inorganic substances in our wastewater. Okay. We do it through introduction of various microbes which can break down biological matter. Okay. What we do is we introduce oxygen so that those microbes can break down the this biological or organic matter, and the broken down material settles at the bottom, and the clear water we get at the top. Now this is what actually happens conventionally in the wastewater treatment plants. But this is a new technology where, in addition to that biological treatment, we use ultrafiltration. Means just like our reverse osmosis at our homes, we use membranes through which we force the water so that even the finest of the particles are removed, and then they also settle down, okay, and we get very clear water. So this is basically used in what? Wastewater treatment technologies, membrane bioreactors. Okay, let us move on to the next one, that is from 2023. With respect to, now again, if you see, this is something, if you have done this and you had come across membrane bioreactors, then you would have focused on that question. Why? Sorry, because you would have focused on that question because this is a similar type of question. With respect to role of biofilters in recirculating aquaculture system, consider the following statements. Biofilters provide waste treatment by removing uneaten fish feed. Biofilters convert ammonia present in fish waste to nitrate. Biofilters increase phosphorus as nutrient for fish in water. Now see this. What we are trying to achieve here is role of biofilters, means through biofilters what we are trying to achieve in a recirculating aquaculture system. What is recirculating aquaculture system? Means in an aquaculture pond where we are not just discarding the wastewater, we are treating that wastewater and bringing that back after degrading the toxic waste out of it. Okay, toxic waste can be waste products generated by fish, or it can be the uneaten fish feed. Okay, so biofilters, again, as I said, this question was what? Membrane bioreactors, biofilters. So these type of questions, once you start getting a hang of these, that this is something that is important, this is something that is asked by UPSC, then your mind will obviously go on these substantial aspects. Okay, so biofilters, if you see, as I said, provide waste treatment by removing uneaten fish feed. This is something desirable, so this will be done. Biofilters convert ammonia present in fish waste to nitrate. Yes, these aquatic animals produce ammonia as waste, like we produce urine. Okay, so present in fish waste to nitrate. Biofilters increase phosphorus as nutrient for fish in water. Now, increase phosphorus as nutrient for fish in water. Phosphorus is something that is required mostly by plants, and living organisms, or if we say not living organisms, means we humans or other animals receive phosphorus through plants, mostly through plants. Okay, or mostly through plants, or if we say through eating, by eating different, means as carnivore as or as omnivore, we can get phosphorus by eating other animals also. Okay, means other within the food chain, means if a carnivore is eating a deer, from there also it can get phosphorus. So biofilters actually do not produce anything. Their role is to remove excess nutrients, and phosphorus is something, it is already saying it is a nutrient. Okay, so biofilters role is not adding that nutrient, its role is removing that nutrient. So this will be wrong, and this 1 and 2 will be correct. Answer will be B, only 2. Okay, now let us move on to the next one. Yes, you can see here the fish tank feed. There are two type of this mechanical filtration, then pump tank through biological filtration, nitrification of ammonium ions into nitrate. Okay, now let us see the next question. This was a very important one. This was a very easy one. If you focus on such things, it becomes very easy for you. Okay, Volbachia method is sometimes talked about with reference to which one of the following? Controlling the viral diseases spread by mosquitoes, converting crop residues into packing material, producing biodegradable plastics, producing biochar from thermochemical conversion of biomass. Now, Volbachia method, let me explain what it is. Okay, Wolbachia is a bacteria. It is a bacteria that is introduced into the Aedes aegypti mosquito. And once introduced, what it does it either means there are two ways it can affect the Aedes aegypti. What is actually Aedes aegypti? Let me also tell if you don't know. Aedes aegypti is the mosquito that spreads dengue. So introduction of Wolbachia bacterium into this Aedes aegypti mosquito is done to prevent spread of dengue. Now it happens in two ways. One is when Wolbachia enters, which bacterium enters a mosquito, what it leads to when there is a mating between female and male mosquito, the eggs that are produced are unhatchable, means they won't hatch. So population will decline. Also those Aedes aegypti mosquitoes which are infected by Wolbachia, they will be less potent. They won't be able to infect somebody with the dengue virus. Okay. So actually this makes what as the correct answer? Controlling the viral diseases spread by mosquitoes. Okay. So this is what is Wolbachia method. As I said, Wolbachia in the Aedes aegypti mosquito bites dengue infected person. Mosquito injects blood with the dengue virus. Wolbachia blocks the dengue virus. As I said, it blocks the Wolbachia. Wolbachia blocks the dengue virus. Dengue infected mosquito with Wolbachia bites another person. That person does not get dengue. Another thing is eggs do not hatch due to cytoplasmic incompatibility. This is important. This is an important term. Means Wolbachia is injected into dengue vector. Then Wolbachia infected mosquito, when it mates with the wild vector that has not been infected, and if they produce eggs, these eggs won't hatch. Okay, so we are introducing Wolbachia bacterium and we are getting the benefit that the dengue virus does not spread or the population goes down. Okay, now let us see the next question is, this was a very important difficult question, but if you understand it properly, then it becomes easy. Genomics is something that we must know as a basic concept in biology. Okay, so the question was, aerial metagenomics best refers to which one of the following situations? Collecting DNA samples from air in a habitat at one go, understanding the genetic makeup of avian species of a habitat, using airborne devices to collect blood samples from moving animals, sending drones to inaccessible areas to collect plant and animal samples from land surfaces and water bodies. Now let us first understand what is metagenomics. Now what you see here is a pond. You see here a heap of sand or a rock. You see here intestine. Okay, what these are three different, if we call populations. Some population of microbes are living here, some population of microbes are living here. Population means consisting of multiple species, and we already know in our gut also there are multiple microbes. Okay, now metagenomics means understanding the complete genetic makeup of an environment of a population. Okay, that is clear. DNA, we are collecting the DNA of complete population and we are doing DNA sequencing and we are trying to find out what are the different kind of species in that population. Okay, so that is metagenomics. Now if you see this aerial metagenomics, collecting DNA samples from air in a habitat at one go, means we are collecting all the DNA, and this does not mean that we have to collect all the air in that environment. Okay, a sample of some water from a pond, a sample of some air from a vicinity, let's say a rainforest, that will be more than sufficient, because living organisms shed their DNA continuously. Okay, and within a sample of air or within a sample of water, all the DNA is there. So rather than focusing on each an animal, trying to find out which animals are there in this population, what we do is we collect a sample, single sample, and then we analyze it. Okay. So aerial metagenomics is clear. Metagenomics is clear. Answer will be A. Answer A. Okay. Next one is microsatellite DNA is used in case of which one of the following. Now this is important. Again, I am saying if you go and search from where UPSC asked this question, these were in Indian Express. That year, both these were in Indian Express. Okay. But then again, there are millions of things that come in Indian Express. So that is why PYQs are important, because you need to understand how the UPSC examiner is thinking, what UPSC examiner is going to focus on. So microsatellite DNA, what is this? Now microsatellite DNA are short tandem repeats. Okay. What are these? Now let me make you imagine something, okay, to understand this. Now let us consider that there are 10 books and each consists of 200 pages. 195 pages are almost same. There are only 5 pages in each book which are different and which actually differentiate those 10 books from each other. Okay, in most simpler terms, microsatellite DNA is those 5 pages. Those part of our DNA, part of our complete DNA, that part of our complete DNA that actually tells us apart from each other. Because within a species, if you are means we are humans, we are animals of the same species, so there is a lot in our DNA that is similar. Okay, so in order to tell us apart based on our DNA, we use microsatellite DNA, and that is why answer is going to be A. Studying the evolutionary relationships among various species of fauna. As I said, microsatellite DNA is something that differentiates each of us. Okay. Stimulating stem cells, not true. Promoting colonial propagation of horticulture plants, not true. Assessing the efficacy of drugs by conducting series of drug trials in population, not true. Okay. It is what it is true for. It is true for A. Studying the evolutionary relationships among various species of fauna. Okay. Understood what is microsatellite DNA? Now let us move on to the next question. Consider the following. Now again, biofilms, biofilter, membrane bioreactors. I cannot emphasize the importance of PYQs more. Okay, you focus on so many things which are not important, and you actually throw away diamonds which are PYQs, and you search for something elusive, something that is going to come that year, and actually, no doubt, UPSC might not repeat that exact question, but there is an aptitude which you develop by doing PYQs. Okay, and as far as I understand, if you are watching this video diligently, then you are developing a number of concepts also. Okay, you won't forget these PYQs again. You won't forget the concept behind these PYQs again. Okay, and if the similar concepts are repeated, you will be able to solve questions. Consider the following statement. Biofilms can form on medical implants within human tissues. Biofilms can form on food and food processing surfaces. Biofilms can exhibit antibiotic resistance. Now let us see what is biofilm. Now this is a biofilm. Biofilm is what? When millions and millions of microorganisms come together into a community, creating, an extracellular matrix. We call it an extracellular matrix. Like when they are alone, they are a single microorganism, but when they come together, they are not just a heap of microorganisms. No, they create a colony, in a matrix in which the different substances, means not only the organic matter which the organic or inorganic matter which the microbes are made of. No, they create a colony, they create a matrix of substances by releasing those substances, and these substances or this matrix protects them. Okay, they can even go into some sort of hibernation within these colonies because now they feel protected. Okay, now once they are within these colonies, they are very difficult to dismantle, they are very difficult to treat with antibiotics. Okay, and they form at various places. They can form at various places. They can form on the implants in our body. They can form on food particles, and whenever they form, they become more difficult to treat with antibiotics or kill with antibiotics, because now they are not just alone, and not only alone, they are in numbers and they have formed a protective layer over them. That protective layer, along with these microorganisms, we call it as the biofilm. Okay, now you see this. So this biofilms can form on medical implants within human tissues, yes. Biofilms can form on food and food processing surfaces, yes. Biofilms can exhibit antibiotic resistance. As I said, they become difficult to treat using antibiotics. So answer will be D, 1, 2, 3, correct. Okay. Consider the following statements in respect to probiotics. Now, means once you start getting, start reading PYQ, start understanding PYQs, UPSC gives a hint, okay, that some things that are going around you, probiotics, probiotic curds are there, probiotic milks are there, probiotic drinks are there, okay, then those questions, those type of concepts become more and more important to you, okay, and UPSC can any day repeat that question either or a similar concept. Okay, so consider the following statements in respect of probiotics. Probiotics are made of both bacteria and yeast. Let us see. Probiotics in the organisms in probiotics are found in foods we ingest, but they do not naturally occur in our gut. Probiotics help in the digestion of milk sugars. Now first let us understand what are probiotics. Probiotics are the friendly microorganisms. Okay, now when you were a kid, you must have heard somewhere that if you see your curd, the curd that we make from our milk, if you see that curd under your microscope, you will not be able to eat that curd again in life. Why? Because there are so many microbes in that. But mind it, those are not harmful microorganisms, those are friendly microorganisms. So similarly, there are many substances, many foods, many types of eatables in which there are such this beneficial microorganisms. Okay, so they are made of both. Bacteria means those probiotics microorganisms can be bacteria, they can be yeast. This is true. Okay, the organisms in probiotics are found in foods we ingest, but they do not naturally occur in gut. This is wrong, because we already know that in our gut we have beneficial bacteria, okay, beneficial microorganisms. Probiotics helps in the digestion of milk sugars. Yes, definitely. There are probiotics, there are this microorganisms which help in digesting milk sugars, and they are in fact very beneficial for those people who are lactose intolerant. Means their digestive system cannot handle milk products, and actually what they cannot handle is the milk sugars. Okay, so this is true, this is true, this is wrong. Correct are 1 and 3. So answer will be C. Let us move ahead. This is probiotics, live microbes that provide health benefits when taken in sufficient amounts. Now this is an additional concept you must know. Prebiotics. Prebiotics are food for the microbes that provide health benefits. Okay, means if you eat this food, your gut flora, we call it the gut flora, the probiotic bacteria that exist there, the flora will love this food. Okay, and they will grow more and more, and they will provide benefits to you. And benefits range from even to mental health mental health benefits, immune system benefits, okay, not only the digestive system benefits, okay. So probiotics, prebiotics. Next, this is important, this is very important, I will explain this. The question was in the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. Very factual question and very conceptual question. If you don't know different types of vaccines, you will never be able to solve this question. Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. Sputnik 5 vaccine is manufactured using vector-based platform. Covaxin is inactivated pathogen-based vaccine. Now, before solving this question, let us first understand what is mRNA vaccine, what is vector-based vaccine, what is inactivated pathogen-based vaccine. Now, vaccine, you need to understand, vaccine is a simple concept of telling our body beforehand or training our body beforehand about an antigen which we believe may attack the body in future, okay. Like, let's say if we talk about polio virus, what we do is we train our body, because our immune system has the capability to remember things, and then produce the antibodies or the killer immune bodies or T cells, which can actually kill the viruses, okay. But for that we need to train our immune system beforehand. Now, training means, as I said, how does our immune system works? Means our immune system remembers that this was the antigen that attacked the body, and this is the way we have to identify it and we have to destroy it, okay. Now, if our body is not trained or it is experiencing some antigen for the first time, so there are chances that our body won't be able to destroy that antigen, okay. So to give our body that memory, what we do is we introduce that antigen, means that virus, that bacteria, in somewhat ineffective form, so that it does not affect our body, but at the same time our immune system develops a memory, means it develops a capability to fight those antigens, okay. Now, there are ways to do that. The most primitive, or the most primitive I won't say, means the most traditional ways to introduce inactivated pathogen, means you weaken the virus itself. Means, let's say we are making a polio vaccine, do you weaken the polio vaccine itself, introduce in our body. Now, what will happen, since it is weakened, the polio virus is weakened, it won't affect our body much, okay. It might cause some fever or some headaches, but apart from that it won't cause much damage. But what will happen is our body's immune system will develop and this resistance towards it, okay, because our immune system will have the memory how to fight it. Our immune system will have the tools to fight that in the future when the actual virus attacks us, okay. Now, there are other ways to do it. Now, this is something that is sometimes a little dangerous, because at times what happens is, though it does not affect that weakened virus, it does not affect the human which is taking that vaccine, but when we excrete or urinate, what happens, this virus can become strong. If it escapes our body and finds another host, it can become strong, and it can lead to means in any case, what we are doing is we are somehow weakening it, and we don't know how much we weaken. Or even if, let's say, the person whom we are giving to, let's say his immune system is compromised, then also it can become means even the weakened virus can become a threat to him or her, okay. So we stepped further. What we said, now let us not send the whole virus into the body, let us send the part that is the most important part. Like we see in the coronavirus, the spike protein is important, so let us send that spike protein in the vaccine. But thing is, now we will need something to carry that spike protein, and what we call that is the vector. Now vector can be a virus also, vector can be a bacteria. Okay, not the bacteria or virus that is going to harm our body, some other bacteria which has been designed in such a way that that virus or bacteria or part of that virus or bacteria will carry that spike protein, let's say, but will not harm our body. Now if we move even more technology means advancement in technology, what we do is we do not send this bacteria, this antigen, in any sense. Means we are not sending it weakened, or we are not sending the spike protein. What we send is the mRNA. Okay, the mRNA means the piece of information that is needed to create that spike protein. Okay, so now the mRNA reaches the body, that spike protein is created, immune system develops, and that spike protein then becomes ineffective. Okay, so these are different levels of advancements in vaccine development. mRNA is good in the sense that it can be mass produced once we have decoded what actually is going to harm the body, and once we have the complete sequencing of its RNA. Okay, now the question was that Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA. No, this was not the case. Okay, this was actually a vector vaccine, Covishield. Okay, Sputnik was again a vector vaccine, and Covaxin was an inactivated pathogen based vaccine. Okay, so answer is B, 2 and 3. So B, 2 and 3. Now you understand what are different types of vaccine. And so now when these concepts come in front of you while you are reading, you need to focus on these concepts. Okay, now let us see. Which one of the following statement best describes the role of B cells and T cells? Now, how many times UPSC has asked questions related to immune system? So many times I had to explain you about immune system. Now, B and T cells are related to immune system only. They protect the body from environmental allergens. No. They alleviate the body's pain and inflammation. No. They act as immunosuppressants. No. They protect the body from diseases caused by pathogens. The antigens which cause diseases, B cells and T cells protect us from them. Answer is D. Now what are B cells and T cells? B cells and T cells are the warriors of our immune system. T cells are really something that, we call it the cell mediated immunity. Cell mediated immunity means T cells, there are some T cells which we call as the killer cells, which actually kill the virus or the bacteria. Okay, B cells are a little less, this killer in this sense. Means what they do is they either inactivate or they either mark that antigen so that other types of immune cells can destroy them. Okay, so B cells and T cells, again we are talking about immune system. So I think it is clear. Okay, now let us see. Now B cells, I attack insiders outside the cell, I attack infected cells. Means T cells can even destroy our own cells which have been infected by viruses or bacteria, okay, or the antigens. Now consider the following statement. DNA barcoding can be a tool to assess the age of plant or animal, distinguish among species and look-alike, identify undesirable animal or plant materials in processed foods. Now we came from microsatellite DNA. This is a similar concept. DNA barcoding is what? Barcoding in our shops is for what? To identify any inventory item. This is a biscuit packet. This is a ketchup bottle. This is a kurkure packet. There is a barcode which we flash the light on, and the barcode is red. So barcode is about what? So similarly, DNA barcoding is about identifying, means differentiating one species or one this member of one species from another. Okay, so assessing the age of plant or animals is not related to DNA barcoding. Okay, it is about differentiating. So answer will be 2 and 3, means D. Clear? So that is why I am saying it is so important to understand PYQs. It is so important to understand the concepts behind them. Okay. Now in 2021, with reference to recent developments regarding recombinant vector vaccines, consider the following section. Now, I already explained you vector vaccines in this previous year. Okay. Now this was in 2021. Now once you get a hang of it, as I am saying, you will be very, very comfortable in clearing UPSC. Okay. Now with reference to recombinant vector vaccines, consider the following. Genetic engineering is applied in the development of these vaccines. Bacteria and viruses are used as vectors. I already told you these are used as vectors. This is correct. Genetic engineering is definitely applied because when we are making a vector vaccine, recombinant vector vaccine, what we are doing? We are combining two different organisms, we can say. Okay. Means we are, let's say we are making this vaccine for, let's say, corona. So we'll have a vector and we will have the spike protein. Okay, so both will be true. Answer will be C. Clear? Recombinant vector vaccines. Okay. Now let's just read this. Viruses, bacteria or cells can be created in lab which carry DNA coding of surface proteins from virus or bacteria. These harmless hybrids can be injected into the body to cause an immune response to the viral surface proteins without making you sick. Okay, I have explained this all already. In the context of hereditary diseases, consider the following statements. Passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization of egg. A child inherits mitochondrial diseases entirely from mother and not from father. Now this is very important. This is a question about 3-parent baby, and this question itself UPSC has asked twice. First statement is passing on mitochondrial diseases from parent to child can be prevented by mitochondrial replacement therapy either before or after. Now you need to understand what is happening here. What happens in mitochondrial related in this genetic disease is that females have, means most of the DNA that we receive, mitochondrial DNA that we receive is from our mother. Okay. Now if our mother has defective mitochondrial DNA, what will happen? We will be inheriting that disease. Now one way to do, there are two ways to do it. Okay, what you can do is, now you will need, now this is why we call it 3-parent baby, because 3 people will be involved. Father will be there, mother will be there, then there will be a second woman or the third person, means there will be a second woman. What we'll be doing is, there are 2 ways as I said. In the first way, what we can do is, we can remove we can remove the nucleus from the this donor, and we can remove the mitochondria DNA from our mother, and then we can fuse those 2 eggs together, and then we can fertilize the egg with the sperm. In this case, what will happen? The nucleus, which contains most of the DNA, will be from the original mother. The mitochondrial DNA, which is only a very small part, will be from the donor. The egg then will be fertilized. Okay. So here what is happening? Whatever is happening before the actual fertilization. Clear? Okay. In the different scenario, what we can do is, we can first fertilize both the eggs, then we can do the transfer. Clear? Okay. So both ways are possible. We call them as maternal spindle transfer and pronuclear transfer. Clear? Okay. So this can be done before also and later also. A child inherits the mitochondrial disease entirely from mother and not from father. This is, as I said, is true. This is also true. So answer will be C, both 1 and 2. As I said, this you can see, mother, father and the donor. Donor comes together. Donor's nucleus is removed. The parent's nucleus is inserted. Okay. and the healthy mitochondria comes from the donor. Okay. Bolgard 1 and Bolgard 2 technologies are mentioned in the context of, now this is important, Bolgard is guarding from bollworms. This can be as simple as that. Means this was it actually. Bolgard 1 and Bolgard 2 are what? These are two varieties of BT cotton which were produced by Monsanto. Monsanto now is, I think, Bayer. Okay. So Monsanto is a multinational company which this created this two types of this BT cotton varieties. Bolgard 1. Bolgard 1 was effective at that time, but soon it was realized that it was not effective against all types of bollworms. So Bolgard 2 was introduced with new genes. Okay. So if we talk about this, this is related to what? Developing genetically modified crop plants. So answer will be B. Okay. Next question is, consider the following. Now this again, as I said, if you understand these concepts, now let me just first read this. Consider the following: bacteria, fungi, virus. Which of the following can be cultured in artificial synthetic medium? Now what people will say, everything is possible in science, just mark as D. This is wrong. Virus cannot be created in artificial and synthetic medium. Artificial synthetic medium means completely devoid of life. As I said earlier in this class itself, that virus is nothing but a piece of information. It requires a host cell to replicate itself. So in a completely artificial or synthetic medium, virus cannot be cultured. Only bacteria and fungi can be cultured in an artificial synthetic medium. So answer will be 1 and 2A. Now you are understanding what is the connection, what is the importance of doing PYQs. All these concepts, you know, your half of the work is done. Even 60%, not even half, more than 50%. Next question is, consider the following statements. Adenoviruses have single-stranded DNA genomes, whereas retrovirus has double-stranded DNA genomes. Common cold is sometimes caused by an adenovirus, whereas AIDS is caused by retrovirus. Now let us understand what is this single-stranded DNA and double-stranded DNA genomes. Okay. Now, adenovirus, retrovirus. These are two different types. As I said, virus is what? It is just a piece of information. Now, what type of piece of information? Information can be in the form of DNA. It can be in form of RNA. Retroviruses carry that information in the form of RNA. So this is wrong. Okay. In a sense, this is also wrong because adenoviruses have double-stranded DNA genome. Okay. And retroviruses have single-stranded RNA. Now let me repeat this. As I said, viruses are nothing but piece of information. Now information can be carried in two forms, either in the form of DNA or RNA. What retroviruses do is, they have that piece of information in the form of RNA. When they enter the host cell, what they do is, they do reverse transcriptase. That is why we call them as retroviruses. Okay. What they do is, they first, with that RNA, they create the genetic information, then they integrate with the host cell, and then they do the replication. Okay. That is the case with all the, that is the case with HIV. HIV is caused by a retrovirus. But our common cold, which is caused by this viruses such as rhinoviruses, that is caused by adenovirus. Okay. So this statement is wrong. This statement is correct. Answer will be B, only 2. Okay. Next is ACE2. The term ACE2, I have already discussed this. I discussed this but if you had known ACE2 in 2020, I think this was ours, or 2021. If you had understood ACE2, then monoclonal antibodies question will be very easy to you. Okay. ACE2, as I said, ACE2 is what? Let me see. Okay. Let me tell you. ACE2, I told you, just try to recall. I told you that How coronaviruses attack. There is a spike protein on their surface which attaches with ACE2 that is located on the surface of our cells. Okay, so if you see this, genes introduced in genetically modified plants, no. Development of India's own satellite navigation system, no. Radio collars for wildlife tracking, no. Is really in the context of spread of viral diseases. Why? Because ACE2 is a protein on the surface of many cell types. It is an enzyme that generates small proteins by cutting the large protein angiotensin. The SARS-CoV-2 virus binds to ACE2 like a key being inserted into a lock prior to entry and infection of cells. Clear? Now this everything is connected. Coronavirus, corona times were so old, so old in the sense compared to 2025. We heard about monoclonal antibodies in 2021, 2022, 2020, but UPSC asked in 2025 in the context of Nipah virus. But the concept remains the same. Clear? Okay, so answer will be D, spread of viral diseases. Now let us see 2020. In the context of recent advances in human reproductive technology, pro-nuclear transfer is used for. Pro-nuclear transfer I mentioned while discussing three-parent baby that there are two ways: maternal spindle transfer and pro-nuclear transfer. So pro-nuclear transfer is related to what? Prevention of mitochondrial diseases in offsprings. Answer is D. Clear? Next is consider the following statements. Genetic changes can be introduced in the cells that produce eggs or sperms of a prospective parent. This, let us see, a person's genome can be edited before birth at early embryonic stage. Human-induced pluripotent stem cells can be injected into embryo of pig. Now, genetic changes can be introduced in the cells that produce eggs or sperms. Now, what I tell you, let me first show you the question in 2017. Now, this was a question in 2017. The question says, what is the application of somatic cell nuclear transfer technology? Now, if you had done this question, if you have understood what is somatic cell transfer technology, then that question have been very easy for you. Now, why? Because there are two types of cell in our body, somatic cells and germline cells. Somatic cells means the cells which do not produce gametes. Okay, all means our skin cell, that is a somatic cell. Okay, but there are cells in our body which produce gametes, means eggs and the sperm. Now, if you make a genetic change in a somatic cell, what you end up is, when you make a change in a somatic cell, you end up changing that cell or that type of cell in that organism only. Like, let's say there is some gene editing done in my skin cells. Now, if I have an offspring, those changes won't be transferred to that offspring because the changes have been done in somatic cells. But if these are done in the cells that produce eggs or sperms, what are these cells? Germline cells. Germline cells. Then it can be introduced, genetic changes can be introduced in this, and that will be replicated in the offspring also. Okay. Now, after that, if you do before that, that is fine, that is also correct. Now, after a person, that embryo has been created, then also a person's genome can be edited. Okay, so this is also correct. Human-induced pluripotent stem cells can be injected into the embryo of a pig. Now, this is very important. Pigs are used a lot in laboratory because in a way they, in terms of size of heart, in terms of size of lungs, they are similar anatomically to humans. Okay, so scientists, what they do is, it means they are thinking about creating organs, creating similar organs. So they are focusing on the research on pigs, and this is part of that, introducing pluripotent stem cells into the embryo of can be introduced in the embryo of a pig. Okay, so all the three statements are correct here. So let us see now. Now again, vaccine. What is the importance of using pneumococcal conjugate vaccine? As I said, UPSC keeps on repeating themes. Okay. Now this is another type of vaccine. We have talked about recombinant vector vaccines. What is pneumococcal conjugate vaccine? This was in news. Now pneumococcal conjugate vaccine. Now this is important. Conjugate here means that we are, creating, means there can be a vaccine where we are just introducing a part of, let's say, that antigen, so that there is, an immunity develops. Okay. But in case of conjugate vaccine, we add something more to that vaccine, okay, to help our T cells develop a better memory about that antigen. Clear? A long-term memory, a more lethal memory, a more potent immune response to that antigen. So pneumococcal conjugate vaccines are used to do that. These vaccines are effective against pneumonia as well as meningitis and sepsis. Yes. Dependence on antibiotics that are not effective against drug resistance bacteria can be reduced. Okay, this can be definitely done, because if we are able to control pneumonia, meningitis, sepsis, then less and less antibiotics will be used, and that will reduce the dependence on antibiotics. These vaccines have no side effects and can cause no allergic reaction. Now this is wrong. As I said earlier also, when we are introducing vaccines in our body, we are introducing some kind of pathogen, then there are chances that our body will react to it. There will be fear, might be headache, nausea, vomiting, and all that. So 1 and 2 will be correct. Answer will be B. Okay. Now let us see. Now as I said, this means two categories: conjugate vaccine and polysaccharide vaccine. But polysaccharide vaccines were not too effective in infects, so we created conjugate vaccines which have long lasting protection, reduces pneumococcal transmission, and better indirect protection. Okay. Which of the following statements are correct regarding the general difference between plant and animal cells? This is important. This is basic biology. Let us see. Plant cells have cellulose walls while the animal cells do not. Okay, plant cells do not have plasma membrane unlike animal cells which do. Mature plant cells has one large vacuole whilst animal cells has many small vacuoles. Now you first need to understand that all eukaryotes have plasma membrane. Okay, so whether animals, whether plants, all have plasma membrane. So plant cells do not have plasma membrane. This is not true. In fact, apart from plasma membrane, they have a cell wall. Why do they have cell wall? Why do they have vacuoles? Means this is also means now, since I have revealed this, plants have cellular walls while animals do not. This is correct. Mature plant cells have one large vacuole while animal cells has many small vacuoles. This is correct. Now I will explain why. Because you need to understand plants do not have bones like us. To maintain structural integrity, they require cell walls and vacuoles. Why? Because vacuoles are something, means in case of animal cells, vacuoles are not that important. But in case of plant cells, they are very important, because cells store water, cells store pigments, cells stores, that water is not only important for plant survival, but also for ensuring structural integrity. Why? Let me tell you, give you an example. Let's say I do not have any bone in my body. Okay. So if I have to stand straight, there must be something that keep me straight. Okay. And that is the pressure inside the plant cells. We call it the turgor pressure. That is inside these vacuoles that keeps the plant straight. And that is why cell wall is needed. Cell wall ensures that there is an equilibrium between these inside pressure and the outside force. So when there is an equilibrium, the plants stand straight, the cells won't burst. Okay. Now you know that if we do not let's say we do not water our plants for long, they wilt. Why do they wilt? Because the turgor pressure which I am talking about, that is lost. If we give too much water, then also they wilt. Why? Because the turgor pressure is too much and the vacuoles burst, or the cells burst, the cell walls burst. Then again they wilt. Wilt means murjha jana. Okay. So answer will be 1 and 3 C. 47. 48. This you can see, plant cell have such a large vacuole. In case of this, these are very small in form of lysosomes, centrioles, and these small vacuoles. Okay. This have a cell membrane. Apart from that, there is lysosomes. Sorry, cell wall. Okay. Now let us move on to 2019. The question was, the question was, with reference to recent development in science, which one of the following statements is not correct? This is very difficult question, but if you understand concepts properly, then it will be easier. Functional chromosomes can be created by joining segments of DNA taken from cells of different species. Pieces of artificial functional DNA can be created in laboratories. A piece of DNA taken out from an animal cell can be made to replicate outside a living cell in a laboratory. Cells taken out from plants and animals can be made to undergo cell division and laboratory petri dishes. Cells taken out from plants and animals, we already see so in another question that in synthetic media we can create different cells. A piece of DNA taken out from an animal can be made to replicate outside a living cell in a laboratory. Yes, this can be done. Pieces of artificial functional DNA can be created. Yes, it can be done, because DNA is nothing but a piece of information, a sequence of information. So this can be done. But functional chromosomes, you need to understand, chromosomes are not just about information. Chromosomes have a particular structure. There is a telomere. Okay, there is a centromere. Means creation of a chromosome functional chromosome is not possible till now in laboratory. Okay, synthetic DNA can be created, in vitro DNA replication can be done, cell tissue culture can be done, but functional chromosomes can be created by joining segment of DNA taken from cells of different species. This is not correct. Okay, so answer will be A, because UPSC has asked about which of these is not correct. Let us move ahead now. RNA interference, this is again a very, very interesting, and again the same concepts are being repeated. Once you understand this concept, this will be very easy for you. RNA interference technology has gained popularity in the last few years. Why? Now let us first understand RNA interference. What is this RNA interference actually? RNA, when we talk about RNA, what we are doing is, what is happening. You need to understand almost every function in our body helps through gene expression. Means there is some information stored in our cells, in our DNA, that what has to be done when something has to be done, means how some, let's say how some protein has to be produced. Okay. Now that information is stored in our DNA, but to make it effective, to bring it into use, that information has to be read. That is the job of the RNA. Okay. That information is read, and then that information is used to create proteins by ribosomal RNA. The ribosomes, we call it the protein producing factory of our cells. Okay. So DNA is a static information. RNA is the working information that we copy, we use it, transfer it, and then we create the proteins. Okay. Now RNA interference means we are interfering with this mechanism of information transfer or information exchange. Let's say we want something not to happen inside an organism. So we interfere with that RNA mechanism to achieve that goal. Okay. It can be used in developing gene silencing therapies. As I said, if you want to stop something from happening, some gene expression you want to stop, you want to silence the gene, then this can be used. It can be used in developing therapies for the treatment of cancer. Definitely, if we can, cancer is about what? Cancerous growth is a cell that has gone on an uncontrolled spree of reproducing itself, and that has become uncontrolled. Okay, so if we want to stop that, we can silence that part of the cell. It can be used to develop hormone replacement therapies. We will see this. It can be used to produce crop plants that are resistant to viral pathogens. Definitely this can be done. Okay, as I said, it can be used to interfere with the normal functioning of a gene. Now this is crucial. It can be used to develop hormone replacement therapy. Now you need to understand, we are not producing anything here. Okay, we are just stopping the expression of a gene. We are just interfering with the mechanism. So we are not producing anything special. So we cannot produce or develop hormone replacement therapies with RNAi technology. So answer will be 1, 2 and 4, that is A. Clear? Now let us see which of the following are the reasons of occurrence of multi-drug resistance in microbial pathogens in India. Genetic disposition of some people, taking incorrect doses of antibiotics, using antibiotics in livestock farming, multiple chronic diseases in some people. Now what is microbial this resistance? If we allow microbes to get habitual to the antibiotics, there is a saying, na, what does not kill you makes you stronger. That applies in this case. Means if we are, means you must have also heard that if you are taking antibiotics, make sure that you complete your course. Why? Because all the antibiotics are created in such a manner that when you start taking them, that course will finish off all the microbes for which you are taking them. Okay, but if you stop them in between, what happens is the microbes are not killed, they are just wounded. Okay, and those means not actually wounded, but I'm using layman terms. They are wounded, and when they rejuvenate themselves, they now know how to fight that antibiotic. Okay, they are much stronger what they used to be. Okay, so when we are exposing the microbes to the antibiotics and are not actually killing them, we are creating a breed of microbes which will not be affected in the same way by those antibiotics in the future. Okay, so this can happen in how many ways? Taking incorrect doses of antibiotics, I already said. Using antibiotics in livestock farming, means if you are using in livestock farming and let's say it somehow gets into soil through, let's say, excreta of the livestock, or let's say we are eating the livestock like poultry, and our microbes or the microbes in our environment are subjected to those antibiotics but not in the amount which is suitable to kill them, then what will happen? They will get habituated and they will develop resistance to it. Okay, so both these are correct. This is not correct. Genetic predisposition of some people, this is not something that is important. Why? Because I didn't say anything like that. It does not depend on me. It is a matter between antibiotic and the microbe. I am just the person who is taking the antibiotic. My genetic disposition does not matter here. Multiple chronic diseases in some people, means in some way, let's say if we have chronic diseases, we will be taking more antibiotics, but this is not a direct reason. Okay, so 2 and 3 will be answer will be B. Now let us see what is Cas9 protein that is often mentioned in news. Now again and again we are coming across questions related to gene editing. Okay, now Cas9 is what? When we are editing genes, we need something. When we are editing something, we need some scissor. If we go in very common terms, we need a scissor to cut the gene, okay, so that we can introduce or remove some part of it. and that is actually what is Cas9. It is a molecular scissor. A molecular scissor used in targeted gene editing. Answer is A. Okay. Now let us move on. This is the Cas9. Which of the following statements is not correct? Hepatitis B virus is transmitted much like HIV. Yes, this is true. Hepatitis B virus is transmitted like HIV through sexual intercourse, through infected syringes, through blood transfusion. Hepatitis B, unlike hepatitis C, does not have vaccine. This is wrong because we have hepatitis B vaccine in India itself. Globally, the number of people infected with hepatitis B and C viruses several times more than those infected by HIV. Yes, this is true because HIV is very lethal. There is, no such data. Means it is, it is a common knowledge that HIV is something. Yes, it is dangerous, but is not as widespread as hepatitis. Some of these infected with hepatitis B and hepatitis C viruses do not show the symptoms for many years. Yes, this is true because the symptoms can take time to show. So the wrong statement is B. Answer is B. Okay. Now let us go to 2018. With reference to genetically modified mustard developed in India, consider the following statements. Now this is very important. People are confused a lot about GM mustard. People think that it is something like BT cotton. It is there to create some type of resistance against some pest or insect. No, it is not the case. Let me explain you, then we will be able to easily solve this question and you will gain an insight into what GM, genetic modification in plants, is capable of doing. You need to understand that in case of mustard, we were not trying to edit in a way to create some kinds of resistance. We were trying to create, a cross-pollination mechanism in mustard. Because what is the issue with mustard is that mustard is a self-pollinating plant. Means it pollinates by itself. Within the same flower, there are male and female parts, and then the pollination takes place within there only. Means there is the self-pollination takes place within that plant itself. Okay, but since we wanted to develop mustard varieties which can have high production, we wanted to create hybrid varieties. So what we did is we created mustard in which we stopped self-pollination and there was cross-pollination, so that when there is a pollination with different varieties of mustard, we can develop hybrid varieties. Clear? Now this will be easy. GM mustard has genes of a soil bacterium that gives the plant the property of pest resistance. As I said, this was not correct. GM mustard has genes that allow the plant's cross-pollination and hybridization. This was correct. Okay, so 2 is correct. GM mustard has developed jointly by IARI and Punjab Agriculture University. Now this was not correct. This was developed in Delhi. Okay, this was developed in Delhi University, University of Delhi. So answer is 2 only B. Sorry, B. 2 only B. Now let us move forward. Consider the following: birds, dust flowing, rain, wind blowing. Which of the above spread plant diseases? Now this is an easier question. Birds, if there are certain microbes or something that can attach to birds feathers or birds legs, then they can be transmitted. Dust can blow those these particles, the plant disease which spread plant disease. Rain can wash down and bring them to other plants. Wind can also do that. Because this was an easier question, it does not need that much explanation. So answer will be D. Okay. Now let us see. Next one is, consider the following statement. Again, this was easier. In tropical regions, Zika virus is transmitted by the same mosquito that transmits dengue. Now this was in news, and it was known that it is transmitted by same mosquito. Now, as I said, now this was question on Zika virus. Then there was a question Wolbachia. So once you understand this, then you will start focusing on these concepts. Sexual transmission of Zika virus disease is possible. Yes, it can be transmitted sexually. Okay, both statements are correct. C, both 1 and 2. Okay, let us see next one. With reference to agriculture in India, how can the technique of genome sequencing, often seen in news, be used in immediate future? Now, what is genome sequencing? Genome sequencing is simply sequencing of our genome. Genome means the complete genetic information. Means in this case it's about plant. If we have the complete genetic information, what can be done? What can be its application? Let's see. Genome sequencing can be used to identify genetic markers for disease resistance and drought tolerance. Genetic markers like the microsatellite DNA we talked about. Okay, microsatellite DNA are also types of genetic markers. Can be used to identify genetic markers for disease resistance and drought tolerance in various crop plants. Definitely, if you know the complete sequence, that we will know what part of that genome is responsible for what, and we can develop that. This technique helps in reducing the time required to develop new varieties of crop plants. Definitely, if you have the complete information, we don't have to do hit and trial. We can do exactly what is needed. It can be used to decipher the host pathogen relationship in crops. Again, as I said, all the information related to our life is contained in the genome. Once we have the genome, all this is possible. So answer will be D, 1, 2, 3. Okay. What is the application of somatic cell nuclear transfer technology? This is important. Somatic cell nuclear transfer technology is about what? Somatic cell nuclear transfer means somebody is using one of my cells and it is transporting somewhere else. Okay. What do we usually do with these cells? We try to clone. Okay. We try to create clones. The sheeps, the buffaloes that we have cloned, that is based on somatic cell nuclear transfer technology only. Means normally how reproduction happens through germ cells. Okay, as I said earlier, there are two types of cells, somatic cells and germline cells. Apart from germ cells, means when we talk about germ cells, we do not clone that offspring. Means in human beings, germ cells leads to reproduction where there is one female and one male. Okay. In case of somatic cell nuclear transfer technology, there is cloning, means exact copy of that organism is created. Okay. So it is used in what? Production of reproductive cloning of animals. Answer C. Now this is somatic cells, somatic body cell with desired genes. This is what is done is nucleus fused with denucleated egg cell, means we denucleate. This is the nucleus. This is an egg cell from which the nucleus has been removed. Within this, we enter the nucleus. Means from the somatic cells, let's say the skin cell, and then we create the clones. Okay. Now let us move on the last year, 2016. In the context of developments in bioinformatics, the term transcriptome, sometimes seen in news. Now again and again I have talked about transcription. The first question that we discussed, that virus cannot affect the transcriptional activity in the host cells. That is about transcription, and now we are almost at the end. Again, the same concept related to transcriptome. Transcriptome is what? A full range of mRNA molecules expressed by an organism. I talked about transcription in case of RNA mechanism. Okay, so transcriptome is what? It is the full range. All other options are not related to it. It is related to the full range of mRNA molecules expressed by an organism. Okay. Now let us see. This is the last question. 10 years, this is the last question asked in biotechnology and health by UPSC. Which of the following statements are correct? Viruses can infect. Again, look at the irony. The second question that we discussed at the start of the video, the statement was viruses cannot affect bacteria. Here question is virus can infect bacteria, fungi, plants. After 10 years, UPSC repeated the same statement. So that is more than sufficient to tell you the importance of PYQs. Okay, so in this case, answer will be D, 1, 2, 3, bacteria, fungi, and plants. Okay, now, so this is done from my side. So in this video, we have completed last 10 years PYQ of science and technology related to the topic of biotechnology and health. Next 3 videos also I will be posting as soon as possible. But apart from that, we will be covering all the other topics or subjects also. Polity, next video I think will be on polity. Then there will be video on CSAT. CSAT videos are going to be very important because almost 10 years and there is so much to learn in CSAT through PYQs, because CSAT is something that has evolved a lot, and the questions that come in CSAT, UPSC CSAT, are something that you do not find anywhere. Okay, so CSAT PYQ classes will be very, very important. This was from my side. I hope you were able to gain something out of it, and I know you were, but still it is my responsibility to remain or skeptic about things so that I can improve. So take care everybody, and we will be coming up with more such videos. By I think 3rd January we will try to cover all the PYQs. Okay, take care everybody.